These aren't even the redwoods. These are mostly like ponderosa pine and Douglas fir. There are some cedar species, and they're just enormous. And there are actually, for a long time, it was sort of a mystery as to why in this one place you have these species of trees that are so big, because it kind of defies physics, because they have to lift water all the way from their roots to their crowns every day, and that's a lot of work. So, why did evolution favor these giant trees? But it did, and they actually know more about it, but we're not going to go into the details right now. So, the place we're going to talk about is the Andrews Experimental Forest, and that would be, let me pause this. So the place we're talking about is the Andrews Experimental Forest, and not only are these trees big around, they're also incredibly tall. And how do you study something that is that incredibly tall? You at some point in middle school did a unit on rainforests, right? What do you call the upper region of the rainforest? It kind of exists in layers. What's that top layer called? Canopy. Yeah, it's the canopy. And the same terminology applies to any forest anywhere. So if you think there's a bunch of stuff going on up in the canopy, how do you study it? Okay, so you can stay on the ground and just look up through glass.
Helicopter.
You can use a helicopter. How do you get close to a tree in a helicopter? What happens? Okay, close to a tree in a helicopter with a long lens. How else could you get up there and study the canopy? Climb it, which is scary and awesome. We're actually talking about trees here that are like the height of 25-story buildings. Figure out what animals live up there and put tracker cameras on them, put a go cam on the whatever. It's a possibility. Throw. Okay. Actually, one of the things that was developed to study the canopies in places like this, old growth canopies, and there are some that actually exist in the rainforest as well, are what are called, I have a really good picture of one of these somewhere. They're what are called canopy cranes. So it's actually a construction crane. There it is. It's actually a construction crane with a very long, this is called the boom. This is the tower. And this little car, that little tiny speck, can travel in and out along that boom, and it can also go up and down. So it can basically go to any place within that area. And so they can get researchers, they can get biologists right up into the treetops. So now we'll go ahead and start our story. In our story, we actually have some biologists and the author in one of these canopy cranes. Baker, our Argonaut, that's the person who's actually collecting samples, murmured, "Okay, Mark, we're done here," into a microphone pinned to his lapel. And we almost instantly plunged straight and smoothly down, then eased to another stop deep in the midst of the old-growth canopy. The gondola was nearly resting on the limb of a western hemlock, and we were suddenly enveloped in a verdure that obscured the surrounding mountains. Verdure is greenness. With the wind suddenly blocked, the air was noticeably warmer. The gondola and any researchers aboard it can, in fact, travel not just the breadth of that 6-acre circle, but up and down to any point within a cylinder. Adding this third dimension means that their access to the canopy comprises a research cylinder of some 58 million cubic feet. Jerry Franklin began promoting the installation of a crane in the early 90s based on a firm belief that the canopy of the old-growth forests remained one of the most important but least explored natural systems on Earth. In fact, the oldest trees here began growing, probably after a fire, just about the time that Columbus landed. In fact, the trees here are so old that they serve as the foundation and the structure upon which an entire airborne ecosystem thrives. Their boles, that means their trunks, and limbs have existed here for so long, they serve as substrates for other forms of life. Shaw goes so far to suggest that the old-growth canopy is an ecosystem directly comparable to one of the most important habitats in the ocean, where living organisms support not just themselves, but provide the structure on and around which a host of others grow or hide or feed. What do you think he could be talking about in terms of the marine ecosystem? A structure in the ocean made up of living things that provides habitat for other things. Who here saw Nemo? What do you think? Coral reefs? Says Shaw, the medium is air rather than water, but essentially it's the system driven by space and light, just like the marine environment. To my mind, the old-growth canopy is very much like a coral reef. Nevertheless, here at the height of winter, it was not much of a day for locating songbirds. That's what they were up there for. In fact, we were able to hear or see only 4 species. A morning in spring would yield 24 or more, from the giant pileated woodpeckers to tiny swifts. But I had come especially to get a close look at something that might seem more mundane: the moss-like lichens that abound here in the canopy. Especially near the top of the canopy, we'd seen a profusion of so-called pendulous lichens, meaning they hang like pendulums. Long, light green masses of tendrils that hang from branches like narrow, pointy beards. Now, deep in the canopy, and we're going to show some of these, deep in the canopy, in a habitat that is shadier and moister, Baker pointed to the very lichen I had come to see. Lobaria oregania, and that is it. And that's like flakes and sheets of this lichen sort of hanging off of a tree. Virtually indistinguishable from the forest floor, the lobaria were growing in great clumps everywhere on limbs and boles here. Folded leafy masses of living tissue growing thick like scalloped layers of lime green icing on a cake. Baker had already shown me swaths of browning needles that had been damaged by a parasite called dwarf mistletoe. Who knows what mistletoe is?
The kissing plant.
The kissing plant! You kiss under it at Christmas. It's actually a parasitic plant. It climbs up trees and it sticks its little roots into them and it sucks their nutrients out, and it can kill entire trees that way.
And why do you kiss people under it?
I don't know. Old superstitions, probably derived from like Europe 5,000 years ago. But that's what we get. That's what we have left. So yeah, it's this parasitic plant that kills trees. So he had already shown me swaths of browning needles that had been damaged by a parasite called dwarf mistletoe. It intrudes into the very core of branches and saps nutrition from the tree, reproducing and moving through the canopy via form of rocketry. It can shoot its sticky seeds as far as 50 feet. A first reasonable guess might have been that the green leafy lobaria were parasites too, sapping critical nutrients from the trees. Because these are hanging literally on every surface of the tree. It's like mold on a loaf of bread. They are just everywhere. One of the first breakthrough bits of research in the Andrews showed that these unassuming organisms, the same sort of mutualistic fungi algae that so fascinated Eugenius Warming a century ago, have much to do with why such magnificent trees thrive here at all. So a quick word about lichen. Lichen, this thing here, is actually 2 species that are living so closely together that they cannot live apart or they'll both die. It's an algae and a fungus. And the fungus breaks down nutrients and gives the algae a place to live, and the algae photosynthesizes and provides food for the fungus. So it's what kind of relationship? Mutualistic. And actually I just realized I said that in the text. But yeah, it's a mutualistic relationship, but there are lots of kinds of lichen. Okay, in fact, it was because of hints from Andrews that lobaria might serve a vital role in the ecosystem, hints that came in part from an old miner's lamp in a plastic garbage bag, that scientists first ventured into the canopy. It was 1969, and botanist Bill Dennison was in the Andrews forest, not looking up at the canopy, but where he thought he was supposed to be looking, down at the ground. One of the first members of the Andrews research team, Dennison had begun, even before funding arrived from the International Biological Program, to try to develop a strategy for what he had already agreed would be his role in the whole ecosystem study. Let's see. Dennison soon met Forest Service ecologist Jerry Franklin and volunteered to become part of the core team that proposed to the International Biological Program the plan to develop a model for how an old-growth forest ecosystem worked. They didn't really understand how the whole system worked. It's like a giant machine with a billion moving parts, and we don't know what they all do in relationship to one another. I was assigned to the dirty rotters, he says with a booming laugh. His initial role, simply put, was to establish how dead organisms and detritus broke down in the old forest, and especially how the critical nutrient nitrogen cycled through the system. As he surveyed the forest, he quickly realized that when it came to nitrogen, he confronted a major problem. There was no obvious source that could provide the critical nutrient in a form that living animals and plants could use. Any gardener knows, says Dennison, that nitrogen is something you've got to have if you want healthy plants. After carbon, he says, and of course hydrogen and oxygen that make up water, nitrogen is the material of greatest need if you're going to try to grow anything. Living things in the ecosystem need nitrogen to form structural proteins and vital enzymes. Fortunately, nitrogen is abundant on the earth, nearly to the point of being ubiquitous. It means everywhere, and comprises about 78% of the what? Nitrogen comprises 78% of the, take a deep breath, atmosphere. Yeah, most of the air around us is nitrogen. Yet few living things can use it directly, and instead must encounter it in the form of nitrates or nitrites. Thus, plants rely on specialized organisms and ecosystems to fix nitrogen. That is, convert it to ammonia and other compounds, which in turn can be oxidized into nitrates and nitrites by specialized bacteria in soils. Typically, bacteria that live within nodules formed by certain kinds of plants, including legumes like beans and peas and locust trees, are responsible for fixing nitrogen in the air. In the forests of the Northwest, red alder trees play such a role. Nodules on the roots are jammed with nitrogen-fixing bacteria called Rhizobium japonicum. But alders normally appear only in bright sun and in areas such as stream banks, or as an early successional stage in the recovery of a burned or otherwise disturbed woods. The Andrews old growth had no red alder, nor did there seem to be any other prominent nitrogen fixers. Bacteria in the soil do provide some ammonia by breaking down amino acids in detritus and dead plants and animals. But based on Dennison's calculations, there simply didn't seem... bacteria in soils do provide some ammonia, but by Dennison's calculations, there simply didn't seem to be enough nitrogen from that source to sustain an entire forest for centuries. So basically, it's like you're seeing somebody who's living in a big fancy house and driving a big fancy car, but they don't seem to ever have any money coming in. How are they sustaining that lifestyle? How are these gigantic trees, I mean, seriously, gigantic trees, look at them. How are they surviving? How are they thriving? How are they growing without some big source of nitrogen? And these are just some more lichen. We'll get to those in a second. We were left with a really big question. Where was all the nitrogen coming from, said Dennison. A quick chemical analysis showed, surely enough, there was plenty of nitrogen available in the soil, but there was little evidence that nitrogen was being fixed anywhere in the system. So where are they getting the nitrogen? It's this huge mystery. It's like, again, like I said, someone's living in this palace and driving, a fleet of Cadillacs, but there doesn't seem to be any money coming in. How are they doing it?
Robbing banks.
Well, okay, there's an analogy there. Yeah, so maybe they're robbing banks. Maybe the money here is coming, maybe the nitrogen here is coming from some source you would never expect. Dennison said that he found himself collecting from the forest floor loose bits and scraps, and sometimes large sheets, of a foliose lichen, which he identified as Lobaria organia. Clearly, it had fallen from the old-growth canopy. The stuff was everywhere, as he puts it. He was still perplexed by the lack of nitrogen fixers in the system, and he began to wonder if somehow the ecosystem's nitrogen fixers were not in the soil, after all, but in the sky. Was it possible that a lichen growing in the canopy could be playing the role of nitrogen fixer, then raining that nutrient down to the soil? By chance, Dennison found himself with an easy way to check. Harold Evans, a prominent botanist and the university's first science elected to the National Academy of Sciences, occupied the office next to Dennison's. Evans had recently pioneered a new technique for identifying the enzymes linked to nitrogen fixation. If exposed in a closed chamber to the gas acetylene, the stuff you weld with, those enzymes will convert the gas to ethylene, which is the gas given off by fruit, by apples and bananas and pears. Dennison applied Evans' technique. He had no need to acquire expensive equipment, for Dennison was a skilled gadgeteer. He still owned an old acetylene miner's lamp he had acquired when he had explored caves years ago. Once widely used as a light source for everything from automobile to headlights to dangerous flaming lights in coal mines. In such a lamp, water drips onto carbide, which reacts to produce acetylene, a bright-burning gas. He promptly rigged the lamp to produce a steady stream of acetylene and injected it into his impromptu chamber, a clear plastic bag filled with lobaria tissue. After Dennison carefully vacuumed the gas from the bag and analyzed it, he found that in fact it had converted to ethylene. Lobaria indeed appeared to be a powerful nitrogen fixer. Armed with that evidence, Dennison spent the spring of 1970 continually looking up, wondering how he could find out what was going on with how he could find out more about what was going on with the symbiotic organisms in the unexplored canopy. How much lobaria was up there? How and when did it grow? And significantly, was there some link between the age of the trees or its forest and the abundance of its lichen? One thing was for certain. Dennison was not going to be able to answer any of those questions without looking in detail at the canopy itself. And when he says these trees are covered in lichen, this, believe it or not, that's a tree trunk. Can you see the tree?
Yeah.
No, you can't actually. All of this stuff is something like lobaria. It's stuff like this. And then hanging on top of it, this is old man's beard. This is what's called wolf lichen. And that tree is literally completely covered in other species. There's not an inch of tree bark that is exposed to air. It's covered. And those lichen, which are themselves a symbiotic organism, are doing a huge service for the tree. So the tree is giving them a place to get up into the atmosphere, and they're providing nitrogen for those trees. They fix the nitrogen. Their tissue becomes full of nitrogen. And then when they drop off the tree in chunks, that nitrogen is available in the soil. What kind of relationship is that? Mutualistic. They're both benefiting. And as I said, if you ever get a chance to go to the Pacific Northwest, go. The very last time that I was out there, I was hiking up into the National Forest, and it was winter. I went over Christmas break, and there had been a storm. And, I mean, you know how branches come down in a forest in a winter storm, heavy snow. So the ground and the road, I was on this little Forest Service road, were just littered with these downed branches from way up high. And they were all just covered in this stuff. It was just everywhere. It's the most amazing thing. And I actually brought home a good bit of old man's beard. It's fascinating stuff. Okay, so talking about forests like this is a really good lead-in. And look at how just absolutely rich and wet. Can you see a single inch of image of sky or anything that isn't a growing living thing?
Yeah.
Where?
Yeah, there at the top. See some sky right there.
Now I just see lights, but I still see trees. I mean, it's really, it's dense.
I don't think that
It's a formerly living thing, and it's in the process of decomposing, and it's covered in living moss, and guess what's inside it? Billions of bugs and bacteria breaking it down. These are incredibly rich ecosystems. Okay, so what we're going to do is we're going to talk briefly about two last concepts that tie into this. So if you would be so kind, I guess it might be 3 slides. We're going to talk, maybe it's 3 slides. We're going to talk real briefly about species richness and species diversity. Species richness, species diversity. So I'll do my normal and read the slide and then shut up while you make sure you have everything in. We can measure things about communities because we've been talking about communities and all these relationships that exist in communities. But there has to be something we can measure about the overall community, and in fact, there is. And the two big ones that we tend to measure are species richness and species diversity. And you know what it means if you're rich. You've got lots of whatever it is that we're measuring in. You can be rich in friends. You can be rich in money. You can be rich in land. You can be rich in chickens. You could be rich in, fill in the blank. I think right now I'm rich in dog fur in my house. And what does it mean if we talk about diversity? Differences. Very often we think about diversity and we think about variety, having a variety of types of people. In this case, it's a variety of types of species in an ecosystem. So we'll first start off talking about that idea of species richness.
I didn't take that slide. It's only richness and
Okay, so the number of species that a community contains is its species richness. This is just down to counting. It's like counting money or counting chocolate chip cookies or counting anything else. Sheer flat out number of species. And species richness tends, this is a generalization, to get higher as you go towards the equator. Species richness tends to get lower as you move away from the equator. So why might this be? Why would you have more species as you get closer to the equator and fewer as you get farther away? What else is there more of towards the equator? No, actually there are a lot of land masses at the equator.
Oh, there's a lot more.
Actually, the northern hemisphere has more land masses than the southern hemisphere.
There's more rain.
More rain in some places, but we also have some deserts along the equator.
The equator is like the rainforest.
There are a lot of tropical rainforests around the equator. Yes, absolutely. There's a whole belt of rainforests around the equator, so there's a lot of moisture. What else is there? What does the equator get more of than any place else on the planet? Sun. That's it. Sun. So the equator gets nearly direct sunlight all year long in this band along the equator. We call that band the tropics. Who needs sun to grow?
Trees and plants.
Yeah, plants in general. And plants are the bottom of all the food chains. Every single food chain, the bottom of it is plants. So if you have more plant species, you can have more herbivore species. If you have more herbivore species, you can have more carnivore species. So if we think about that, if we have plant species, 1, 2, 3, 4, okay, lots of plant species. That gives us room for more, well, let's see, make these guys blue. Herbivore species. Okay, and if we've got more herbivore species, it makes more room for carnivore species. And having lots of those makes more room for what we'll call decomposer species. So those plants are getting energy directly from the sun. Oops. Because plants are getting energy directly from the sun. Herbivores are getting energy from them. Carnivores are getting energy from the herbivore species for the most part. And decomposers are getting energy from all of them. The more energy there is in the system, the more of everybody you can have. The more energy there is in the system, the more of everybody you can have. Okay, so the other thing we look at is species diversity. And species diversity examines not only how many species there are in a community, but how many individuals there are of each species. And communities can be stable or not stable. When a community is resistant to change, we say it's stable. And communities with more individuals of more species, higher species richness, tend to be more stable than communities with fewer species. Why would a community that was more species rich be more resistant to change? Let's go back and think about this graphic. So if these are not only species but individuals, every single herbivore that's getting nutrients from a plant is doing a job. Every single carnivore that's getting energy from an herbivore is doing a job. Every decomposer that's breaking down dead stuff is doing a job, and you really want your decomposers to work. If your decomposers are not working, the world is just full of rotting things. Actually, they wouldn't even be rotting. The world just fills up with dead stuff if your decomposers don't go to work. So what happens if you lose half of your herbivore species? Or half of your plant species, rather, and half of your herbivores, and half of your carnivores, and half of your decomposer species. Are all the jobs still going to get done?
Maybe.
Maybe, maybe not. What if you're starting off with a much smaller number and you lose half of those species?
Probably not.
Yeah, and it's harder for your community to recover if you're not getting all of these jobs done. It's much harder to recover. Whoops. So what we tend to find is that if you have higher species diversity, it's much easier to recover from a disturbance. Okay, we're gonna try to do a paper lab on species diversity, species richness tomorrow. And then we're going to talk a little bit about succession. We're not going to do a whole lot with succession. And then we're going to roll into doing your test for this chapter. So I think for most of you, you would prefer to have the test before you go away for 4 days rather than after you go away for 4 days. And then it's a clean slate. You don't have to study over break. You're just ready to go. So we're going to plan on doing the quiz in here on Thursday. And obviously it'll have to be a shorter quiz because we only have a half an hour on Thursday because of Battle of the Classes. So you'll have your study guides tomorrow at the end of class, and we'll do a little bit of what we're going to do, and then I'll turn you loose. Obviously you're updating your vocab as we go along because I'm going to grade vocab probably on Wednesday. I keep saying it. Questions, comments, concerns? Okay.